Good morning, students. Today let us see the topic proteins, properties of proteins. First of all, let us see what is a protein. Protein is a macromolecule composed of one or more polypeptide chains possessing a characteristic amino acid sequence. It is a polymer of amino acids. You can see the protein molecule and amino acids in this picture. In this picture, you can observe the molecular formula of protein. The term protein is derived from Greek proteo, means primary or holding first place. The term protein was first proposed by Berzelius. Protein is the essential constituents of living cells. Proteins made up of 12% of the protoplasm. They are the body builders. They contain carbon, nitrogen, hydrogen, oxygen and sometimes sulfur. They are constructed largely of amino acids. In this picture, you can see the sources of proteins. Proteins can be obtained from either plant sources or animal sources. From plant sources, we can get proteins from beans, grams, peas, lentils and soya beans, etc. In animal sources, we will get protein from eggs, paneer, meat, milk and fish. Proteins are obtained both from animal and plant sources. The animal sources of protein include milk, egg, meat, fish, liver, etc. Plant sources of proteins are pulses, nuts and cereals. Nutritive value. The nutritive value of a protein is based on two factors, namely amino acid composition and digestibility. All proteins do not contain all the essential amino acids. Only a few types of proteins contain the complete set of essential amino acids. Such proteins are called first class proteins and their nutritive value is high. Almost all the animal proteins contain all the essential amino acids. Example, milk, milk products, meat, fish, kidney, liver, shellfish, soya beans, etc. The proteins which are deficient in any one of the essential amino acids are called second class proteins and their nutritive value is low. Example, cereals, nuts, pulses, etc. Now let us see the properties of protein. Physical state. Most of the proteins are hydrophilic colloids. A few proteins such as insulin, tobacco mosaic virus, etc. are crystalline in nature. Color. Proteins have no characteristic color except chromoproteins. Chromoproteins are colored and the color is imparted by the metallic prosthetic group. Taste and odor. A pure protein is tasteless and odorless. Viscosity of proteins. Proteins are highly viscous in nature. Generally, fibrous proteins are more viscous than globular proteins. Fibrous protein, example, fibrinogen. Globular proteins, example, albumin. Molecular weight. Molecular weight of proteins varies from 30,000 to a few million. Protein molecules are much smaller. In fact, the molecular weight of a typical protein is comparable to that of the smallest nucleic acid molecule, that is tRNA. Typical polypeptide chains have molecular weights ranging from 15,000 to 70,000. The average molecular weight of an amino acid is 110, which means that typical polypeptide chains contain some 135 to 635 amino acids. The molecular weight is determined by ultracentrifuge. Levorotatory. All the proteins are levorotatory. This property is due to the presence of alpha amino acids, which are the building blocks of proteins. Hydrolysis. Peptides and proteins undergo hydrolysis by means of dilute hydrochloric acid or sulfuric acid, alkali or enzymes into their constituent amino acids. Hydration. When proteins are brought into contact with water, protein molecules absorb water and swell up. The polar groups like COOH, NH2 and OH become hydrated. Electrolytes, alcohols or sugars in high concentration will compete for the water of hydration, dehydrate the protein and precipitate it from solution. Next one, coagulation. Most of the proteins are hydrophilic colloids. They are precipitated or coagulated in solutions alkaline to the isoelectric pH by positive ions such as zinc, cadmium, lead, etc. At this pH, the protein has a negative charge. Negative ions combine with proteins in solutions more acidic than the isoelectric pH to form salts. The common precipitants in this group are trichloroacetic acid, tungstenic acid, phosphotungstic acid, sulfosalicylic acid, picric acid and tannic acid. Salting in and out of proteins. The solubility of many proteins is increased in the presence of small concentrations of various neutral salts. This is referred to as salting in of proteins. Salting in of proteins is caused by forces of attraction between salt and protein at low salt concentrations, leading to increased solubility. As the concentration of the neutral salt is increased, the solubility increases to maximum and then starts decreasing, and finally the protein is precipitated. This is referred to as salting out. The salting out of proteins is caused by the competition between the protein and the salt at high concentrations. The salting out of proteins is an effective method of purification of proteins. Oxidation of proteins. Proteins are oxidized by putrefaction processes to form the products such as amino nitrogen compounds, carbon dioxide and water. The bad smell of the dead and decaying bodies is due to the putrefaction of the protein. Amphoteric nature of proteins. In proteins, there are several free amino groups from basic amino acids and COH groups from acidic amino acids. The groups can ionize in solution, producing both anions and cations. Thus, the protein molecule exists as bitter ion in solution. It can combine with both acids and bases. Thus, proteins are amphoteric compounds. Isoelectric point. As each protein contains one free NH2 group and another free carboxyl group, the protein has an ionizing property. Due to the presence of these ionizable groups in the protein chains, the proteins also have some definite isoelectric pH at which they do not migrate in an electric field. At the isoelectric pH, the number of positive charges is equal to the negative charges, giving a net charge of 0. Proteins are cations at pH values lower than the isoelectric pH and anions at pH values higher than the isoelectric pH. Denaturation. Proteins undergo remarkable changes in their solubility, optical rotation and biological properties when they are treated with heat, X-rays, ultraviolet rays, light, alcohol, acetone, aqueous, potassium iodide, urea, detergents, etc. These changes occurring in proteins are called denaturation. The changed proteins are called denatured proteins and the unchanged natural proteins are called native proteins. Color reactions. Proteins give characteristic colors on treatment with specific reagents. These reactions help to identify proteins, hence they are called test for proteins. There are 6 tests to identify proteins: Biuret test, Millien's test, Ninhydrin test, xanthoplerotic test, Heller's test, nitroprusside test.